Assalamu alaikum dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 48. The topic is the citric acid cycle. Discussing agenda for the topic will be conversion of pyruvate to acetyl coenzyme A, and then we will discuss the citric acid cycle, which is also known as tricarboxylic acid cycle or Krebs cycle. Lecture outcome. After watching and listening this lecture, students will know how pyruvate is converted into acetyl coenzyme A and subsequent events with the acetyl coenzyme A. Of course, then acetyl coenzyme A, once synthesized, enters into Krebs cycle or citric acid cycle. Introduction. Now first we will discuss how the pyruvate, which is the end product of glycolysis, converts into acetyl coenzyme A, because the starting point for Krebs cycle is acetyl coenzyme A and the end product of glycolysis is pyruvate. So what are the different steps which leads to the conversion of pyruvate into acetyl coenzyme A? After pyruvate is oxidized, the citric acid cycle completes the energy yielding oxidation of organic molecules. Glycolysis releases less than a quarter of the chemical energy in glucose that can be harvested by cells. Most of the energy remains stockpiled in the two molecules of pyruvate. So in glycolysis, only 2 ATPs were produced, and along with those 2 ATPs, 2 NADH molecules were produced. NADH are the carriers of high energy electrons, and these NADH molecules then start the process of electron transport chain. But the two pyruvate molecules still remains, so more energy is to be yielded from the remaining pyruvate molecules, but it all depends upon the presence of oxygen. If oxygen is present, then complete oxidation of the glucose molecules takes place and all the events are completed. If oxygen is not available, then the pyruvate molecules either enters into lactic acid fermentation or alcoholic fermentation. So here what we are studying basically is we are considering the presence of oxygen in the cell. When O2 is present, the pyruvate in eukaryotic cells enters a mitochondrion, where the oxidation of glucose is completed. In aerobically respiring prokaryotic cells, this process occurs in the cytosol. Now, the prokaryotic cells lack mitochondria, so the process of electron transport chain and glycolysis and Krebs cycle occurs in the membrane of the cytoplasm or within the cytosol. Later in this chapter we will examine other fates for pyruvate. Other fates means when there is oxygen not available, then what will happen to pyruvate? For example, when oxygen is unavailable, or in a prokaryote that is unable to use oxygen. So now we are discussing the fate of pyruvate in cells where oxygen is available, and later on we will discuss the fate of pyruvate in cells which are lacking oxygen or which are unable to use oxygen. For example, they are anaerobes. Now, pyruvate enters the mitochondria through a transport protein and is then processed by a complex of several enzymes known as pyruvate dehydrogenase. You can see here pyruvate dehydrogenase. This enzyme is responsible for the conversion of pyruvate into acetyl coenzyme A. Now there are a total of 3 steps involved in the conversion of pyruvate into acetyl coenzyme A, and all these steps are catalyzed by this multi-enzyme complex pyruvate dehydrogenase. So let's see what are these steps. The first, pyruvate's carboxyl group. Remember, pyruvate is a 3-carbon molecule. So the pyruvate's carboxyl group, already somewhat oxidized and is thus carrying little chemical energy, is now fully oxidized and given off as molecule of carbon dioxide. So the first reaction is the removal of the carboxyl group. So when carboxyl group is removed, then 2 carbon molecule is left. And remember that this is the first time where carbon dioxide is liberated in aerobic cellular respiration. In the second step, the remaining 2 fragment molecule is oxidized and the electrons transfer to NAD, storing energy in the form of NADH. So 1 NADH is produced here, 2 NADH are produced in glycolysis, and remember that the end product of glycolysis is 2 pyruvate molecules. So this process will happen twice. So 1 NADH molecule for each pyruvate. So we can say that the oxidation of pyruvate to acetyl coenzyme A produces 2 NADH for the 2 pyruvate molecules. Now in the third step, coenzyme A, a sulfur containing compound derived from a B vitamin, is attached via its sulfur atom to the 2 carbon intermediate, forming acetyl coenzyme A. So now this molecule can enter into Krebs cycle. Acetyl coenzyme A has a high potential energy which is used to transfer the acetyl group to a molecule in the citric acid cycle, a reaction that is therefore highly exergonic. So that third step completes the conversion of pyruvate into acetyl coenzyme A. You can see here, this is the pyruvate. It's a 3-carbon molecule, and one of the carbon is released, the carboxyl group is released, and it is released in the form of carbon dioxide. Now the remaining 2 carbon fragment gives its electrons to NAD and form NADH, and the remaining portion combines with coenzyme A and form acetyl coenzyme A. So remember, the enzyme involved is pyruvate dehydrogenase, and this whole reaction is known as oxidative decarboxylation, this whole reaction. And where it takes place inside the mitochondria. You can see all the stages are taking place inside the mitochondria. So now we move on to the citric acid cycle. What are the different events which takes place in the citric acid cycle? So it's after the synthesis of acetyl coenzyme A, this molecule enters into citric acid cycle. The citric acid cycle functions as a metabolic furnace that further oxidizes organic fuel derived from pyruvate. So the pyruvate is converted into acetyl coenzyme A, and then acetyl coenzyme A enters into Krebs cycle, where further oxidation of this molecule takes place. In the subsequent slides, we will study the whole cycle. We will summarize the whole process. This is the text portion. Figure 2 summarizes the input and output as pyruvate is broken down to 3 carbon dioxide molecules. So because pyruvate has 3 carbons, so all those 3 carbons are liberated as carbon dioxide molecules, including the molecule of CO2 released during the conversion of pyruvate to acetyl coenzyme A. So during the conversion, the 3 carbon pyruvate molecule loses its 1 carbon, and then it loses its further 2 carbons in the Krebs cycle. So all the 3 carbons of the pyruvate are lost or completely oxidized. The cycle generates 1 ATP per turn. Because there are 2 pyruvate molecules, so it means that 2 Krebs cycles will take place, and each Krebs cycle produces 1 ATP. So it means that 2 pyruvate molecules will produce 2 ATPs. Now this is again a substrate level phosphorylation. Substrate level phosphorylation also took place in the glycolysis, where one of the substrate donated the phosphate group to ADP and formed ATP. Here once again, one of the substrate, one of the intermediates of the Krebs cycle will provide the phosphate group to ADP and form ATP. So remember that in aerobic cellular respiration, 4 ATPs are produced through substrate level phosphorylation, where a substrate provides a phosphate group to ADP, and the major bulk of ATPs are produced in electron transport chain, and all those ATPs are produced through oxidative phosphorylation. Now, in the Krebs cycle, another electron carrier is also produced, which is called FADH2. The reduced coenzymes NADH and FADH2 shuttle their cargo of high energy electrons into the electron transport chain. So 2 NADH molecules were produced in the glycolysis, and then 2 more NADH molecules were produced in the conversion of pyruvate to acetyl coenzyme A, and then in the Krebs cycle more NADH molecules were produced. So these high energy electron carriers, then run the affairs in electron transport chain. Now, the citric acid cycle is also called the tricarboxylic acid cycle or the Krebs cycle. Hans Krebs was the scientist who in 1930s elucidated this cycle. So this Krebs cycle is basically to honor the person who elucidated this cycle. Krebs was the German-British scientist largely responsible for working out the pathway in 1930s. Now let's look at the citric acid cycle in more detail. The cycle has 8 steps, each catalyzed by a specific enzyme. So there are a total of 8 enzyme catalyzed steps in Krebs cycle. Each turn of the citric acid cycle, 2 carbons enter in the relatively reduced form of acetyl groups and 2 different carbons leave in the completely oxidized form of CO2 molecules in step 3 and 4. Now you can see here this acetyl coenzyme A molecule is entering into this cycle, and it is having 2 carbon molecules, and then one of the carbon molecules is both the carbon molecules are liberated in step 2 and 3. Now citrate is the ionized form of citric acid for which the cycle is named. The next 7 steps decompose the citrate back to oxaloacetate. It is this regeneration of oxaloacetate that makes the process a cycle. For each acetyl group entering, 3 NADH are reduced to 3 NAD are reduced to NADH. In step 6, FAD instead of NAD is used and FADH2 is formed. So this is substrate level phosphorylation. The GTP may be used to make an ATP molecule or directly power work in the cell. So there is no specific difference between GTP and ATP, and it is formed in the 5th step of the glycolysis Krebs cycle through substrate level phosphorylation. Now, what is the end product of Krebs cycle? The end product is 6 NADH, 2 FADH2 and 2 ATP. Now remember that because we are considering the complete breakdown of a single glucose molecule, that single glucose molecule ends into 2 pyruvate molecules, and then these 2 pyruvate molecules enters into 2 Krebs cycles. So the total sum is this. If you are considering a single pyruvate molecule, then it produces 3 NADH, 1 FADH2 and 1 ATP. So this is the diagram which I draw to the students a year or 2 ago, and here we will analyze this whole process. Now this is oxaloacetate, and this is the acetyl coenzyme A, which was produced from ATP. from pyruvate. So this is the first step. The enzyme involved is citrate synthetase. So citrate is produced. Oxaloacetate is a 4-carbon molecule, and the 2 carbons in acetyl coenzyme A combines with it, so it forms 6 carbon molecules. And then in the second step there is first dehydration and then rehydration. Water aids again, so it forms isocitrate, which is also a 6 carbon molecule. And this is the step, the third step, where the carbon dioxide molecule is released. So it was 6 carbon molecule, one of the carbon was released as a carbon dioxide, and then the alpha ketoglutarate is left, which is 5 carbon molecule. In the step 4, one more carbon is given out, and now it becomes a 4 carbon molecule. So the liberation of carbon dioxide takes place in step 3 and 4, and then only a single ATP is produced, and that is produced in step 5. And then in step 6, the FAD becomes the carrier of electrons, and it forms FADH2. So a total of 4 oxidation events takes place in Krebs cycle. This is the first oxidation event where NADH is produced. This is the second oxidation event where NADH is produced. And this is the third oxidation event, and this is the fourth oxidation event. So in a single Krebs cycle, 1, 2, 3 NADH molecules are produced, 1 FADH2 molecule is produced, and 1 ATP or GTP is produced. Remember, this is for 1 pyruvate molecule. So it is summarized here. 3 NADH are produced in step 4, step 3, 4 and 8. GTP is produced in step 5, and 3 carbon dioxide are released. This 3 carbon dioxide refers to the 2 which are released here and the 1 which was released in conversion of pyruvate into acetyl coenzyme A. So now the glucose molecule is completely broken down, completely oxidized. But one very important step is left in aerobic cellular respiration, and that step is electron transport chain, in which those high energy electron carriers will deliver their electrons, and it will start the process. Most of the ATP produced by respiration is generated later. Later means in electron transport chain, and the process is oxidative phosphorylation, when NADH and FADH2 produced by the citric acid cycle and earlier steps relay the electrons extracted from food to the electron transport chain. Now let's calculate the number of ATP, number of NAD and FADH2 produced. So in the Krebs cycle, in the glycolysis, in the glycolysis 2 NADH molecules were produced, and then the conversion of pyruvate to acetyl coenzyme A. This process produced one more NADH, and then this acetyl coenzyme A entered into Krebs cycle, and in the Krebs cycle 3 NADH were produced, and 1 FADH2. So this is only for one pyruvate molecule, 4 NADH and 1 FADH2, but we have 2 pyruvate molecules, so 6 NADH, 6 in the Krebs cycle, 2 in the conversion of pyruvate to acetyl coenzyme A, that makes 8 NADH, and when you combine it with the 2 NADH formed in glycolysis, so it makes up a total of 10 NADH. And because Krebs cycle occurs twice for 2 molecules of pyruvate, so 2 FADH2. And also the first 3 steps also yield 4 ATP molecules. So now only 4 ATP molecules are produced, but all of them are produced through substrate level phosphorylation. Now these are the high energy electron carriers, and then they will enter into electron transport chain and they will deliver their electrons, and they will further produce the major bulk of ATP. Now this is yet another explanation of this process. So this is the acetyl coenzyme A, which enters this cycle and it combines with oxaloacetate. It forms a 6 carbon molecule citrate, then isocitrate. The first NADH is formed, and then the liberation of carbon occurs in step 3 and 4. In step 4, one more NADH is produced. In step 5, the only ATP is produced. In step 6, the FADH2 is produced, and then in step 8, another NADH is produced. So 1, 2, 3 NADH are produced, 1 FADH2 is produced, and 1 ATP is produced. So this is the whole process, and remember that at the end the oxaloacetate is regenerated again. So that's why this process is a cycle and not a straight pathway. So this was the end of the lecture. Thank you very much. Allah Hafiz.